Thank you for the introduction. And, this is Zhifeng from WuXi, one of the sponsors. And today, it's my great honor to be here to hear about RNA biology and drug discovery. And I want to present the platform and services developed at WuXi, which can be provided to researchers for their RNA drug discovery. The first slide is the general workflow of RNA targeting drug discovery at WuXi. As a service platform, we are always dedicated to providing a one-stop solution to clients for their target of interest. So specific to RNA, we start from RNA synthesis and characterization. We investigate the folding of your RNA preparation, and we test the integrity and stability. We do shape map to verify RNA secondary structure to ensure the quality, and once confirmed, then your RNA preparation will be applied to be screened using our affinity-based screening platforms to identify small molecule ligands. And those small molecule ligands can be synthesized at WuXi, and we do have a bunch of biophysical binding assays for you to validate those small molecules. And here is our toolbox specialized for screening RNA targets. Our RNA drug discovery is derived from our small molecule library collections, including the specialized DNA encoding library, the fragment library, and the traditional HTS small molecule libraries. And by using zipper DEL selection, by using the biophysical binding assays, and using the ALICE SMS, these libraries can be screened against RNA targets. And our screening platform, particularly the DEL selection, can yield huge data sets with tons of binding information. So we have built an integrated DEL data-based machine learning pipeline to take the full advantage of our huge data set, where those trained models can be used to either validate our screening or to identify novel structures. And we do validate those identified small molecules by using orthogonal assays. We are well equipped to run basically all the biophysical assays and to verify RNA secondary structure and potential ligand binding site. We do shape map. Shape map is a technology where RNA functional motifs can be covalently and selectively labeled by those shape chemical probes. These shape chemical probes can cause mutations during reverse transcription, which can be further identified by sequencing to reflect RNA secondary structures. So now ShapeMap is a very popular service in our RNA platform, and ShapeMap can quickly tell you the folding of your RNA preparation and can predict potential ligand binding site. We can do probing either in vitro or in cells. The turnaround time is about 2 to 4 weeks for you to get the data. And here is a showcase for SMN2, where SMN2 transcript, including exon 7 and the adjacent intron sequences, were folded, and the RNA secondary structure was determined by using the shape map. Our data identified several stem loops, which is consistent with previously published data, and particularly this terminal stem loop 2, which is located at the exon 7 and intron 7 junction with the implication of regulating RNA splicing. Our company started building our first DEL library in 2017, and within 5 years, we are now a major player in this field by providing DEL libraries and DEL library screening services to clients. And here is an overview of our DEL modalities for different screen purposes. And we have classic traditional DEL with up to 90 billion unique DEL molecules. And these libraries have been provided to hundreds of clients to screen their targets, including RNAs. And we also have covalent DEL with either irreversible warhead or reversible warhead. We have microcycle peptide-like DEL with a certain percentage of this library has peptide-like structures. And we are also working on bifunctional DEL to screen for molecular glues or PROTACs. My colleague Alex Satz, he's going to give a presentation on Thursday. And if you want to know more details about those other modalities, you're welcome to join his talk. However, I would like to say DAO selection against RNA targets is more challenging as compared to screening protein targets. This is mainly due to the interaction between RNA and the DNA barcodes of our DAO molecules. A DAO molecule is a small molecule compound covalently linked to a DNA barcode through a long flexible PEG linker. Though, the DNA barcode was designed to be a double-strand DNA followed a 3' single-strand DNA tail. So the reason for this design is to facilitate reverse primer annealing during the PCR amplification. However, the single-strand DNA region can cause significant interaction between RNA and our DNA barcode. So to enable DAO selection against RNA targets, we need to convert the single-strand DNA region to double-strand DNA, and we call it zipper DAO. And here is the data showing that single-strand DNA's motif-specific binding can be mitigated by DNA zipper treatment. And we use riboswitch as our POC, and those RNAs were well folded and screened in parallel with either the warheadless, the single-strand traditional DAO, or the warheadless specialized zipper double-strand DNA DAO. And the sequencing data was subject to DNA binding motif analysis. From these plots, you can see a significant difference between this orange line, which is UP2, that's the traditional single-strand DNA DAO, and the blue line, the ZUP2, that's the zipper double-strand DNA DAO. So this data indicate that the interaction between riboswitch and the DNA barcode can be significantly decreased by the zipper treatment. This is really critical because we want the chemical warhead binding pattern to be really identified in a real DAO selection, and it shouldn't be diluted or interrupted by DNA binding patterns. So with that, we have screened with this specialized zipper DAO, we have started screening some internal RNA targets and identified, quite a few novel ligands. And here is the most recent story for SMN2. So you can see that those cubic scatter plots, we can demonstrate the enrichment of this building block 2 and building block 3 disynsome family, which is significantly enriched by SMN2 at different concentrations. But in the no target control and in the G4 mutant control, this enrichment was either not visualized or dramatically decreased, indicating that the binding between this disynsome family might be specific to SMN2. DAO identified small molecules need to be further validated because they are DNA barcode conjugated. We need to do off-DNA resynthesis, and then we need to validate those compounds in another assay. Given that DAO selection is affinity binding based, so we normally want to suggest another binding assay to do this validation. SMN2 DAO selection identified, a few chemotypes. So we have synthesized some, off-DNA resynthesized compounds, and here is the data for these 13 compounds. These 13 compounds, they were pooled together with a positive control, the risdiplam, and these compounds, they were incubated with SMN2 transcript, and by using the size exclusion chromatography, binders were separated from non-binders and further identified by high-resolution mass spec. So basically, we are using ALICE SMS to validate these 13 compounds. From this data, you can see that actually 8 out of 13 compounds can bind to SMN2, where compound 10, so those highlighted in red, has the highest retention. So we repeated compound 10 at different concentrations. From this binding saturation experiment, we can see that the KD can be estimated to be 2-digit micromolar. Of course, binding doesn't guarantee the biological function. So to further test compound 10, we have done this fluorescence indicator displacement assay, and we can see that compound 10 can actually bind to the terminal stem loop 2, because terminal stem loop 2 overlaps with RNA splicing site. So we further tested, we further did the SMN2 mini gene expression experiment in HEK293 cells. Well, I would like to thank Dr. Mala because she already presented the mechanism of this disease and how this gene was involved in this disease. So I will not go through those details. But we just did the mini gene expression experiment, and we treated those cells with our compounds, and then RNA splicing was determined by using RT-PCR. So from this agarose gel, you can see that the positive control, the risdiplam, clearly showed increased E7 inclusion. And our compound 10, is less effective, but we can still see about 50% E7 inclusion, indicating that our compound 10 may be functional in rescuing SMN protein expression. So I would like to wrap up this SMN2 story here and just bring you a message that our specialized ZipperDel platform is robust to identify small molecule ligands and binding to RNA targets. So in the end, please allow me to highlight our libraries one more time. We have DNA encoded library collections, and our DNA encoded libraries can be split for different business modes. We have DEL-Link, which is covalent library, and we have DEL-Open, which is our support to the academia with libraries free of charge, and we have DEL-Lite, which is a kit. Everyone can buy it, and you can do DEL selection following our SOP in your own laboratory and send your samples back to our facility, and we do simple processing, sequencing, and deliver the final report. And DEL Pro is our most popular service. DEL Pro contains libraries built with WuXi AppTec proprietary building blocks and scaffolds. So DEL Pro has the best diversity and novelty, and DEL Pro is only performed by experienced WuXi selection scientists. And we do care about the quality of DEL molecules. We try to improve our libraries every year to make sure they are more drug-like and can cover chemical space as most as possible. So other than DEL libraries, we have traditional HTS libraries, and here is our 3000 fragment library whose quality is well controlled, with each fragment verified by using LC mass, and all these compounds, they follow the components of the rule of 3. And we also have a 300,000 HTS small molecule library collection. And those small molecule libraries, they were chosen carefully for diversity and drug likeness. And I would like to say we are ready to run those small molecule libraries by using ALICE SMS to screen RNA targets. As mentioned, we have a bunch of biphysical binding assays like SPR, MST, TRIC, and etc. Those assays can be used either to screen RNA or to validate RNA binders. And here is a showcase where our client's messenger RNA was screened against our fragment library. You can see that the hit rate is about 3.6%, and we identify compounds, and those compounds can be further validated in separate SPR experiments. And to better support RNA drug discovery, we are now expanding our toolbox by collecting more and more data from in-house internal RNA internal RNA screening experiment to accumulate those RNA-prone building blocks. And together with knowledge from literatures, we are working to build our own RNA-focused libraries, including DEL, fragment, and HTS. And please, I would like to say, our open access capabilities and our technology platforms can provide quality services and to help our customers to quickly achieve their goals. And we are now trusted by more than 5,950 customers. And I really want to acknowledge our customers. They are our motivation, and their success is our success. So with that, I would like to stop here, and I'm happy to take questions. Thank you for your attention.
Right, questions? So a really interesting application of the DEL. Potentially a naive question here. In addition to the challenge of RNA DNA binding, did you also have to address issues like just the inherent lack of stability of RNA compared to proteins?
Well, this is a frequent question.
Yeah.
So as a service platform, we do have some advantages by checking different database, targeting different RNA targets. Well, if this is a frequent hitter, we are not going to suggest those compounds to our clients. So we don't want to tell our clients, well, this compound also binds to other RNA targets, but we just don't want to suggest that.
I guess I'm more asking also, thanks. In terms of your workflow, right?
Yeah.
Just like the way you handle proteins, you can probably just be, you probably have to be more careful just with RNAs, right? Just because RNAs are just not that stable, right? Is there any any lessons that you have in that area in terms of dealing with RNA stability in the process of the DEL screening?
Yeah, so I would like to say, well, RNA preparation is actually the key to a successful DEL selection. Well, maybe 10 clients come to us with the same RNA targets, but their RNA preparation might be very different. So we have this shape map to verify RNA secondary structure and quickly tell you the quality of your RNA preparation to guarantee the quality. But to be honest, RNA biology now is challenging. I still think RNA biology is challenging because we don't know which RNA preparation or which RNA secondary structure is the most appropriate preparation and can be applied to this screening. So we need more experiments.
Thank you so much. For your DEL libraries, do you hold the compounds without the DNA tags? And if not, how long is the typical period to synthesize them without the tags so that they can go into the ALICE screen to confirm the hits?
Okay, so for DEL libraries, all the compounds, they have to be conjugated with a DNA barcode. So DEL libraries are our primary screening. So after that, every compounds need to be off DNA resynthesized and further validated. It's about 6 to 8 weeks for you to get the off DNA resynthesized compounds. And then we can do ALICE SMS. ALICE SMS is very fast.
Okay, gotcha, thanks.
Yeah, thanks, Zhifeng. Nice talk. So you mentioned the importance of the single-stranded region on DEL to get efficient PCR amplification, but then you zip that up to a double strand. So then how do you unzip it so that you could then efficiently amplify the output?
Yeah, great question. I'm not allowed to disclose too much, but we have to change back to single-strand DNA and do PCR. So there's an additional step between DEL selection and PCR amplification. I'm sorry about that.
That's your unzipping step.
Yeah.
All right, there's another question here on this side.
Hi, nice talk. I know a pretty common strategy with protein DEL selections is you'll incubate the protein with some generalized double-strand DNA. It's usually reported as salmon sperm DNA. Did you ever try, incubating your RNA targets with some kind of double-stranded DNA construct or maybe single-stranded oligos to try to take up any potential DNA binding sites, kind of in the traditional sense? Was that just insufficient? Are those interactions too weak and they fall apart?
I think this is a strategy already published by our competitor, and we don't do that. We just because if you think the interaction is between single-strand DNA and the RNA, so personally I'm thinking which method would be more effective than blocking the whole single-strand DNA region? So that's my answer.
I have a question here. So you mentioned you have a covalent fragment library. Is that also a DEL or is that a non-DNA encoded library?
It's DEL. We call DEL link. Now it's a product. So yeah.
So are the warheads tuned down so they don't react with nucleophiles and nucleic acids then?
It's targeting cysteine, and we have about 6 to 7 electrophiles. And because for covalent libraries, we don't want the compound to be too big. We only include this 2-cycle and 3-cycle libraries there. Yeah.
So that's not something that you'd use for RNA.
It's not. It's definitely not.
Thank you. Other questions?
Hi, so you mentioned using shape on the sample that's given to you to tell if it's like correctly folded. Is that based on like how the client thinks the RNA is folded? And like how do you tell if your sample is folded well versus, maybe flopping around a lot?
So great question. So even I don't have the answer because without literatures, without previous publications, or without NMR data, actually we don't know. We can only say, well, we can see some stem loops, this is structured, and we can also try different folding conditions to see whether the structure is kind of stable. But to be honest, which one is the best, I don't know. Well, just there's no special conditions, just some literatures. Some for other RNAs, we just follow that. And if clients they have very strong experience about their specific RNA, we just follow their instructions. Yeah, just using some sorts. Yeah.
Right. One last question.
Hi, thanks for your great talk. Kind of piggybacking off the previous question, do you ever see differences in DEL's hits that come out if, let's say, you fold the RNA under different conditions and import those conditions to your DEL screen?
I think so. Normally, because unless this is internal, RNA target, we are going to try different folding conditions. We can tell you a story, but for clients, normally they don't want to do that. They may just try to use one folding condition and try to see whether they can find clients find potential ligands. Because anyway, this is kind of fee based. They have to pay some money to do this. But for internal RNA DEL selection, we do try different folding conditions. But no matter how, because we don't know which one is the best, so we don't know which one can actually give you the most physiological relevant binders, and we don't know whether those compounds they can be really functional. So it's a very tough question. I think with more and more experiments and with more and more clients come to us, we will have more experience, and maybe I can answer your questions better at that time.
All right, let's thank the speaker. And after the coffee break,